afternoon, everyone. This is Kathy McGlynn. I'm a past president of the Northeast Aquatic Plant Management Society, and I welcome you to the first session of our winter webinar series in 2024. There are a few house rules I wanted to give you before we start the presentations, so we'll just go through that quickly. So just giving you a little bit of background about the Northeast Aquatic Plant Management Society, our mission is to assist in the management of aquatic vegetation, to provide for the scientific and educational advancement of members, to encourage scientific research in all facets of aquatic plant management, to promote an exchange of information among members, and to extend and develop understanding in the discipline. Our members are from academia, government agencies, private consulting firms, herbicide manufacturers, and lake associations. We have annual meetings, annual plant camps, please ask us more if you're interested, and webinars which bring CEUs for folks and offer student scholarships up to $25,000. If you're interested in learning more about the society, please check out the link below. So our house rules for today, the webinar is set to run from 1 to 3. There will be 2 45-minute presentations with a block of 25 minutes at the end for questions and discussion with presenters. Please type your questions into the chat box and the moderator, myself, will read them during the Q&A. Please note that we are unable to offer CEUs for this particular webinar session, but we'll be offering them for session 2. The webinar will be recorded and it will be shared via link on the NEAPMS website. All right, so our presenters today. Our first presenter is Alex Martin from the Nature Conservancy of New Jersey. He'll be talking about Vallisneria, hydrocaritaceae, taxonomic revisions and implications for restoration ecology. Alex recently graduated with a master's degree in ecology and evolutionary biology from the University of Kansas. His research specialty is systematics, phylogenetics, and taxonomy of aquatic plants. Growing up in Minnesota, he didn't pay much attention to aquatic plants except avoiding them when fishing or swimming in lakes, but in 2012, he gained a strong interest in growing aquatic plants as a hobby, which eventually propelled him into a new career path. In early 2023, he was hired by TNC, or the Nature Conservancy, as a freshwater science specialist. His work includes data collection and analysis of water quality parameters in the Pollenskill River and floodplain reforestation stewardship. Our second speaker is Dr. Katja Engelhardt from the University of Maryland Center for Environmental Sciences, Appalachian Laboratory. She'll be talking about Vallisneria genetics and propagation in response to herbicide testing. Dr. Katja Engelhardt studies the connection between biodiversity and ecosystems functions and likes to keep her feet wet while doing her research. She has a passion for conducting actionable science that enhances restoration efforts and builds connections with managers and lifelong learners. Dr. Engelhardt received her PhD at Utah State University, where she studied trumpeter swans and their food resources. She then joined the faculty at the University of Maryland Center for Environmental Sciences Appalachian Laboratory in Frostburg, Maryland, where she's now a research associate professor. Her current research focuses on genetic diversity as a factor in enhancing the restoration success of wild celery, otherwise known as Vallisneria americana, in the Chesapeake Bay and the Hudson River. In support of this goal, she has developed the extensive genetic repository of Vallisneria americana in the Appalachian Labs greenhouse, where she researches propagation and planting techniques. In times of dry feet, Katja loves being active with her husband and 2 daughters, friends, and 2 Siamese cats. So I will sign off now and let Alex begin.
Okay, so I think you should see full screen now, right?
Yes.
Perfect. Okay, so like she said, my name is Alex Martin. In 2023, I graduated with my master's degree from the University of Kansas. And my thesis project revolved around the genus Vallisneria, which is the topic of today's presentation. So a brief overview of the things that I want to cover today will include things like some background information about the genus, including morphological features, systematics, and taxonomy. And then I'm going to cover one restoration project that is currently undergoing in Central Florida. So first, for those of you not familiar, what are Vallisneria and how can we identify them? Vallisneria are monocots, which means they are related to familiar plants like grasses and sedges, and they are categorized as submerged aquatic vegetation. The generalized growth habit can be seen here on the left. They grow leaves from a rosette or a base crown at the bottom of the plant, and they can reproduce asexually by making clones of themselves through stoloniferous growth. They are dioecious, meaning that an individual plant is either male or female. And typically female Vallisneria plants will produce a solitary female flower on a long stalk called a peduncle, although there have been observations where these plants have been observed making female flowers in umbulates, but the reason why this is happening is currently unknown. Pollination with these plants occurs through wind and water movement. In this picture, I'm showing that there's some multiple of these microscopic male flowers resting at the surface right next to the female flower. So the female flowers will sit at the water surface and create a little cavitation in the water that aids in drawing in the male flowers towards the stigmatic surface. And once pollination has occurred, the peduncle will contract and coil and bring the female flower underwater where the fruit will mature and then the seeds will start to form. And despite their lack of insect or animal facilitated pollination, we actually do see some variation in female flower morphology. So in the collection that I had growing in the greenhouse at the University of Kansas, I was able to observe female flowers from Vallisneria australis, Vallisneria spiralis, Vallisneria gracilis, and Vallisneria nana. And then onto those microscopic male flowers. The picture on the left, I'm showing a flower, an individual male flower that I pulled from an immature spathe. So that little structure down there that's enclosed. The spathe contains hundreds of these male flowers, basically like a hermetically sealed balloon. And then once these spathes crack open, it'll release these tiny little male flowers that will eventually float up to the surface. They'll crack open and the sepals will reflex to act like a little flotation device. You can see the pollen grains sitting on top of the filaments right there. And just to see how small these things are, and in that slide on the right, I have a picture of them next to a dissecting pin, and they range from about 1 to 2 millimeters tall. And then with my research, I was particularly interested in leaf color morphology. So pictured here, you can see that there's some major differences in the coloration patterns with different species of Vallisneria. And from what I can assess, it appears that the plants that have these strong horizontal dark striation patterning seem to be restricted to the areas found in the tropics or the subtropics. And then one of the diagnostic features that I was interested in as well was Vallisneria have serrated leaf edges. So some species I noted had these serrations going all the way down the entire length of the edge of the plant, while some of them just had these serrations just at the leaf tip or the apex. And then really interesting is that there are 3 species of Vallisneria found only in Australia that grow along a stem rather than just at that base rosette. There's Vallisneria caulescens, Vallisneria triptera, and Vallisneria rubra. So Vallisneria are found worldwide, although about half of the species within the genus are found within Australia. So here's a map that I pulled from the Global Biodiversity Information Facility. And the little dots that you see here on the map are from both iNaturalist research grade occurrences and also from when an herbarium does digitization of the specimens that they have in their catalog. And then one of the reasons that I chose this genus for my project was that it's relatively understudied when it comes to taxonomy and systematics. So some previous research on Vallisneria includes this 1982 paper from Richard Loden. He was the first to assess species diversity within all of Vallisneria. And so in his research, he noted that there was a lot of difficulty assessing species types by just using vegetative morphological features because they were susceptible to phenotypic plasticity, meaning that the way that these plants look is greatly affected by where they're growing. And so instead, he decided to use just floral traits for his analyses. And at this time in 1982, there was about 9 accepted species of vals. And then after he got done with this paper, he decided, there's only 2 species of valves found throughout the world, with about 4 different varieties within them. And then in 2008, Less et al. Was published, which became the most robust investigation into the genus. They were able to analyze both molecular and morphological data from all the available species that they had at the time, and they estimated the group's first phylogenetic relationships using parsimony and Bayesian analyses. And with that, they ultimately ended up with a much more speciose genus than Loden. And then in 2023, I published a paper within the Journal of Aquatic Botany based on the first chapter of my thesis. And I used molecular data from the new specimens in my study, as well as combining those with sequences that I pulled from GenBank to produce a phylogeny using maximum likelihood. And based on my results, I gave an updated species list. But unfortunately, my master's degree was affected by a global pandemic, seeing as I went from 2020 to 2022. So I was unable to assess genetic information from 2 species that were described at or around Wuhan, China. And currently there's no sequences available on GenBank for either of these. So I was unable to assess the relatedness in the phylogeny to the other Vallisneria species. But however, I was able to get some Vallisneria plants sent to me from Northern Territory, Australia. My collaborator, Dave Wilson, agreed to send me plants. It took about 5 months of paperwork and dealing with the Australian government, but we were able to successfully get a shipment over here. And that ended up in me describing a new species that I'm calling Vallisneria jacobsii. So at the request of Dave Wilson, I've chosen to name the plant after Surrey Jacobs. He's a distinguished botanist who worked at the Royal Botanic Gardens in Sydney. And although I never got to meet Surrey, everybody that I've talked to about him, they speak highly of his character and his accomplishments surrounding aquatic plant research in Australia. And more to the note, within North America, based on molecular data, we continue to see that there's the temperate Vallisneria americana and the subtropical Vallisneria neotropicalis as they're starting to form their own monophyletic groups. So for my research project, I wanted to do some field work to investigate these differences that we're seeing in these extreme locations. So in July of 2021, I was able to go to my home state of Minnesota and collect Vallisneria americana from Square Lake, which is located about 20 miles north of the Twin Cities, near the Wisconsin border. This lake is well known for having very clear water. Secchi depths go up to about 46 feet or 14 meters. The littoral zone of the lake where all the plants are growing goes to about 40 feet deep from what I was able to observe. But due to the limitations of me freediving for plants, I was only able to collect plants from about 10 feet deep or less. The plants that I collected, I brought back to Kansas. I planted some of them in the greenhouse, the common garden that I had going there. And then the others I vouchered within the McGregor herbarium. And then about a month later, in August, I collected plants from I think I lost. Can you hear me again?
Yep, we've got you.
Can you hear me? Oh, hold on. Microphone check.
Yep, you're good.
Okay, perfect. Okay, yeah, my headset went off for a second. Sorry about that. And then in August of 2021, I collected plants from Central Florida. So some of the vales came from Lake Hernando, where we were staying at an Airbnb. And then others came from Little Jones Creek, which is just north of Lake Panasoffkee. It's a spring-fed creek and maintains a temperature of around 72 degrees Fahrenheit or 22 degrees Celsius all year. And the valves here had leaves that were about 3 meters long. So that is to say they were the biggest Vallisneria plants I'd ever seen so far. And then I just want to show you the differences between the Vallisneria that I collected in Minnesota and the Vallisneria that I collected in Florida. So here's a picture showing what they look like after growing together in a common garden environment for about 6 months. On the left are the ones that I collected from Minnesota, and then on the right are the ones that I collected from Little Jones Creek in Lake Hernando. So knowing now that we're trying to push to recognize 2 species within all of North America, the question remains, where are the species boundaries found throughout the United States? Would we expect to see some discrete separation between species with obvious boundary lines formed by a geographic barrier? Or do we expect to see a gradient where there's a large hybrid zone that exists where we start to see these intermediate morphologies between those 2 extremes with hybrids? So this is a project that I would love to work on in the future because understanding species boundaries is really important for restoration projects. So Vallisneria have been the focus of restoration ecology for a while due to their ability to provide habitat for macroinvertebrates, for fish, and they provide a food source for waterfowl and manatees. And they also provide multiple benefits for increasing water quality. So the one project I'm going to cover is the project in Kings Bay, Florida, or Crystal River. So how I came across this project was completely on accident. I originally planned this trip just to go to Florida to collect plants, but seeing as my wife and I were celebrating our 10-year wedding anniversary that year, we decided to make it a dual purpose trip. And then one day my wife brought up the idea that we should go on a clear kayaking tour in the Crystal River. And I said, yeah, let's go. We have a chance to see some manatees and it'll be really cool. And when we got there, I saw that the entire bay was filled with Vallisneria. So Kings Bay is brackish environment, so the freshwater springs pump in millions of gallons of fresh water into that bay, and that meets with the salt water coming in from the Gulf. And then here's just a quick video showing how many valves were in that bay. This is from the side of the clear kayak as it was underwater. And you can see there's so many valves you can't even see the sand at the bottom. So in Kings Bay, there were these large beds of native Vallisneria that were a food choice for migrating manatees that visit that part of Florida every winter. But due to multiple factors, including an increase in salinity from what they call a no-name storm back in 1993, nearly all of the Vallisneria died, and they were quickly replaced by cyanobacteria. The Kings Bay Restoration was formed, and they aimed to remove all this harmful algae and replant Vallisneria that was saltwater tolerant. So with the help of the University of Florida and an aquatic restoration company called Sea and Shoreline, they developed a variety of Vallisneria that they called Rockstar and Salty Dog and used that for the project. But unfortunately, after I collected those and placed them within the data set for my phylogenetic analysis, it appears that they are more closely related to Vallisneria densa and Vallisneria spiralis, which are native to Europe and Southeast Asia. And when you add their sequence data into the Gorham et al. 2021 dataset, it shows evidence that they are the same non-native hybrids that they were finding in their study. These hybrids were first detected in Japan. And then a few years later, they were discovered throughout Florida, and they were believed to have originated from hybridization in aquaria and then subsequently introduced into non-native areas. And based on the conversation that I had with the Florida Wildlife Conservation Commission, it seems that the Kings Bay Restoration Project was looking for Vallisneria plants that were saltwater tolerant. So they went around the coastal areas around Central Florida, and they found one that was growing in brackish water and brought it back to a nursery setup to grow in mass and use for their project by replanting them. So the researchers there at Florida Wildlife Conservation Commission have subsequently published information on using PCR analysis to detect hybrids and other common hobbyist plants within Florida, so to stop them from being released into the wild. And unfortunately, this paper was a little too late. During the preparation for this presentation, I saw that there was another project in Florida that's using these exact same hybrids for their project. And then additionally, right after I gave this talk at the conference, I had somebody come up and tell me, oh, you know, Rockstar, it's being discovered in Tennessee as well. And so this was just a news clipping that I pulled up online about the Rockstar hybrid being found within a reservoir in Tennessee. And so the question is, you know, is this hybrid going to be an issue? Is it going to outcompete native Vallisneria? And at the time, I guess I really couldn't say yes or no, but as I've highlighted down at the bottom, aquatic plant biologists are currently unable to find or detect native species because the rockstar eelgrass is taking over. So my recommendation for anybody that is wishing to use aquatic plants or Vallisneria specifically in a restoration project and wants to avoid possible ecological disasters is to spend a little bit amount of money to get genetic sequence data before replanting from a transplant location. And if it's a resource that you don't have available to you, there are labs out there who would do it for you. The example I'm giving here is Dr. Tippery's lab at the University of Wisconsin-Whitewater. You can send him plant material and they will do the DNA sequencing for you and get you the results back pretty quick. But if you're like me and you have access to the materials to do it yourself, like I did with my project, it's not too difficult. You take so I took plant material, so I just took leaves off of the plants, dried them in silica from 24 to 48 hours, threw some steel beads in with the plant tissue in these epitubes, and then pulverized the tissue. You can either do that with like a mortar and pestle. I used this machine, the archaic machine that's seen on the right. The commercial variant of this machine is called a Geno grinder. We called this the De-annihilator. It was just a reciprocal saw. The Geno grinder is about $20,000 machine. This was about $200 from Home Depot. Made a lot of noise, very fun to use. But it just pulverized the plant tissue. And then that pulverized tissue, I threw that into this Qiagen D&EZ Plant Pro kit to extract DNA. Once the DNA was extracted, I checked the quality and quantity of the DNA using a qubit fluorometer and a nanodrop spectrophotometer. And then after verifying that I had sufficient DNA, I ordered PCR primers for the 3 gene regions that were used in that Les et al. 2008 paper. And then I ran PCR on our thermocycler. And then the amplified products, I verified that I had what I needed based on gel electrophoresis. And then I sent those amplified products out to ACGT Inc. for sequencing. So the sequences get sent back to you in this text format. You can just open it up in like a Word document or a notepad, but then they also send you chromatogram data. So this is just a verification of the sequence data based on they send the sequences through a machine that shines light at every nucleotide, and these peaks that you're seeing at the bottom are just showing like this is a confidence of like how the sequences show up. And in my study, I was sending them some plants that I thought were hybrids, or they were ones that were in the hobby. There's one that was called Leopard Val.
I'm gonna have to plug my headset in.
Just give me 1 second. I'm gonna plug my headset in. I keep losing connection.
We can still hear you, Alex.
Can you hear me again?
Yes, we got you.
Perfect. Okay. So one thing that I noticed from the sequences that were getting sent back to me, this is the chromatograms that I was getting from the nuclear ribosomal ITS region. So this is a region that is inherited by both parents. So unlike the chloroplast regions that I was also sequencing, the chloroplast regions are only maternally inherited. These are inherited. This gene is inherited by both parents. So when we capture this sequence, sometimes we get these single nucleotide polymorphisms that I'm showing at these red arrows. And this is just the difference between the two parental types. It can also be variation within this sequence itself, but previous studies suggest that within this region, if you see multiple areas that are showing this double peaking, as seen here with these red arrows, it's evidence to suggest that they might be hybrids. And so these are the polymorphic sites I was finding within the Kings Bay vowels. And what's interesting is you can actually partition these sequences out to be two sequences by using a process that Don Less called pseudo-cloning. And so you can kind of give a guess of what might be one parent type for this hybrid cross, and then separate that out to form two sequences to try to get a hypothesis for what the parent types are for that hybrid cross. And again, I think that this is a quality control check that should be implemented for restoration projects, especially when you're taking one plant type and growing it and then transplanting it to another location. So I took the sequences that I got from the company, and then I was able to assemble those sequences in CView. And then I also pulled sequence data from GenBank. For phylogenetic tree building, I used IQtree, which is a web software used for phylogenetic analyses using maximum likelihood. So IQtree uses Model Finder to find the best fit model for the data, and I ran an analysis using 100 bootstrap replicates for node support and an approximate Bayes test for branch support. So this is the phylogeny that's was used in the paper. This is using a combined molecular data set with maximum likelihood analysis. And then this was the phylogeny that I used for my thesis presentation, just because I wanted to note which plants I used for my project, as well as some of the hybrids. The reviewers did not want the hybrids within the combined data set analysis for the publication, so those were left out. But you can see the rock star there, that 17 rock stars found within that Vallisneria spiralis and Vallisneria densa cerulata. And so what's the takeaway from this talk, and what did I hope you learned? So there's some species of Vallisneria that have very distinct morphological features that can be helpful for species delimitation when you're, and for a field identification when you're out in the field. Molecular data gives us a good understanding of Vallisneria and how they're related on an evolutionary scale, and it can also be used for identifying species when that morphological data isn't helpful. And then our understanding of species diversity and distribution is getting more refined as we're able to capture more genetic data. And please try to avoid unintentionally spreading non-native plants to areas. Just allocate a small amount of your funds from a project to do some genetic testing. And with that, I thank you for your time, and I'm open for questions.
So Alex, we'll wait to do questions at the end of
Okay.
both presentations.
Oh,
So
okay,
we'll
sure.
go ahead and welcome Katya. If you wanted to start your presentation, that'd be great.
Hello, everyone. Can everybody see this?
Yes.
Excellent. Okay, thanks for inviting me. I'm here at the Appalachian Lab, which is part of the University of Maryland Center for Environmental Science. It's part of the University System of Maryland. I'm here in the Appalachian Mountains, so not in the big metropolitan area, but up in the mountains. And I'm excited to talk to you about Vallisneria americana, genetics and propagation. So first of all, I'd like to just provide a little bit of context. When we think about restoration, at least when I think about restoration, and I know you all do too, being into aquatic plant management, is I think of it as rolling a boulder up a hill like Sisyphus does in the myth. And it's really hard, right? It's incredibly hard to do restoration. It's time intensive, it takes a lot of effort, and it also takes a lot of money. And it's also possible that, you know, you are doing a great job and you're halfway up a hill, and then the political climate changes and your funding gets ripped away and you can't maintain that boulder where it is. And so I feel like the analogy, the Sisyphus analogy, really works well with restoration, thinking about restoration. So I also like to think about restoration in terms of 5 myths. This is something that was published by Hildebrand et al. In 2005. The paper is called The Myths of Restoration Ecology. In that paper, they discuss 5 different myths. This is not at all criticisms of restoration, but it's a way of framing different restoration projects. They're not all the same, of course. So the 5 myths that they describe are the carbon copy, which is we want to do something similar to what we already know, whether it is a habitat that is right next door, we want to copy it, or something that that we used to have and we want to have it again. Field of Dreams is about build it and they will come. So if you build a habitat, then for sure species and individuals will populate this area. Fast forwarding is, well, we might actually build the habitat, but we know that they will not come, or they will come maybe in 20 years, but we don't want to wait this long. So this is where maybe some planting or some reintroductions would occur in the fast forwarding myth. The cookbook is something where we have a protocol, and if we just follow that protocol, then we will be successful. I'm thinking of my daughter, 14 years old, who made Christmas cookies, and she really wanted to make that. These cookies were they're sort of sugar cookies with the Hershey's Kiss in the middle. She really wanted to make those, and she followed the cookbook really well, except for she put twice as much butter in, and it was a disaster. So anyway, a cookbook can be followed and it might not work, or you do just something a little bit different and it doesn't work at all. Command and control, this is sort of what they call, Hildebrand et al. Call the Sisyphus complex, which is, it's sort of the engineering mindset that we can degrade things, it's okay, because we're humans and we're really intelligent, and we can just, with enough effort and enough money, we can make it right again. So those are the 5 myths. Now, however, so then the question is, when we do restoration, can we make it a little bit easier on us? And so that's that top guy right there. Is there any way we can do restoration so that it's not quite so hard? It's easier to get that ball up the hill where we want it to be. So then that leads me to the question of, well, what's the goal of a restoration? So a bunch of people actually have thought about this, of course, and so this is just one definition of a restoration goal. I'll just read it to you. The ultimate goal of ecological restoration is reestablishing self-sustaining ecosystems that will be resilient to future perturbation without ongoing human input. And so what needs to be underlined here is self-sustaining, so that we don't continuously have to put that effort in. And then also be resilient to future perturbation, because the environment changes and we do have perturbations. So we can't just have a little storm come through and then everything is screwed up again. So I like to actually put that Sisyphus picture on its head a little bit, or on the side, and that's where we want to be, right? So we've taken the boulder to where we need it to be, and now it can just stay put there. How do we do that? So that gets me to aquatic plant management, because I think we all have that interest in common today. So the goals for aquatic plant management, this is where I would have opened it up for discussion a little bit, because I actually do like interacting with an audience, but it's not, this menu doesn't work that well for that. But the goals for aquatic plant management are restoring degraded habitat, right? And so when it comes to the myths of restoration, that might fall under build it and they will come, the field of dreams myth. Our goal might be that we want to control invasive species. I think a lot of us are interested in that. Well, here that myth might be the cookbook approach, right? So when we think about herbicide use or any sort of way to control, there is a protocol to follow that is based on science, you would hope, and then and if we just follow it, then we will be successful. Well, we know that that's not necessarily the case, because every environment is a little bit different. But I would think that that restoration approach would follow more the cookbook approach, the cookbook myth. And then another goal of aquatic plant management might be reintroducing species. So maybe a habitat was degraded, maybe a hurricane came through and scoured out a habitat. We've lost all of our individuals from that area. The whole submerged aquatic plant bed is gone. And we would like to have those SAB back. And so here we might think of the carbon copy myth. We want it back the way it used to be, or we might actually want to reintroduce some species to fast forward the process. So maybe they would come on their own. So it could be a field of dreams myth, but we want to fast forward it with reintroducing species. And that then gets me into Vallisneria americana. We've already heard quite a bit about Vallisneria, and I just want to continue on that train of thought. So Vallisneria americana is a very common submerged aquatic plant species in the Hudson River, as well as the Chesapeake Bay, which is kind of in my backyard. Well, it's 2.5 hours away, but so this species, as you know, is in the whole genus of Vallisneria, and many of those species that you see here on the left, which is the phylogeny of it, there's many of them in Australia, and also in Asia, but we do have one, Vallisneria americana, that is very common here in the United States. We also have Vallisneria neotropicalis, which is further south in the southern US. And in fact, actually, we were trying to sample across the entire east coast of the US. And at some point around South Carolina, we actually weren't able to get our markers to perform well in terms of our microsatellites. And so we were thinking that that's probably because it wasn't necessarily Vallisneria americana anymore, at least not purely so, but it could have been a hybrid, or it even could be neotropicalis. So we're talking about Vallisneria americana now, because that's what we have in the Chesapeake Bay, as well as in your backyard, the Hudson River. As Alex already said, the species is just really cool in that it has some asexual reproduction. And so you can have these long strings of plants that can still be attached to each other. And so this is actually the Potomac River here, and you can see there's, I don't know, I've never actually counted them, but I'm guessing probably about 15 plants strung along, and it probably, that clone was even bigger than that. We've actually found clones that are like 100 kilometers apart. They're actually one of the biggest clones that we know of, and the reason for that is that these ramets, the individual plants can actually disattach, and then they can float in the water for miles, kilometers, and then actually root somewhere else. And so we found that especially dominant in the Potomac River. We've also seen it a little bit in the Hudson River. And so these ramets, when they senesce in our climates, the Maryland as well as the New York climate, they, in the winter months, they will put their energy into these underground turions, which you see here on the right-hand side. So the above ground vegetation senesces, and then you end up with these turions. And so what's really great about this is that you can work with this, right? You can actually figure out different genotypes, then you can propagate these different genotypes, and from one plant, you can actually end up getting hundreds of plants over just a few years. Not only do they reproduce asexually, but Vallisneria americana also reproduces sexually. And we've already heard about that, so I don't have to belabor that. We have male flowers that produce these flowers that rise up to the water surface. They of course have pollen in those flowers, and then the female flower sits on the top of the water, and then they come in contact with the male flowers. Then you end up with reproduction, a seed pod is formed, and what you see here on the bottom right is a bunch of seeds which are tiny. So one pod can actually produce like 200 tiny little seeds. So asexual and sexual reproduction is happening with Vallisneria americana. Some of the threats that we have in the Hudson River as well as the Chesapeake Bay is eutrophication, sedimentation, like that's both of those, eutrophication and sedimentation is due to runoff from the watershed. Storms come through. We have salinity intrusion closer to the ocean. We also, of course, have temperature increases, which is especially an issue, starting to become an issue in the Chesapeake Bay, and maybe not quite so bad in the Hudson River since it's cooler up there. Of course, we also have herbivory. There's Canada goose herbivory as well as fish to some extent. And then, of course, we have invasive species, so many different threats. Here on the right, we have a picture, I'm sorry that it's so fuzzy, of Hurricane Lee, which came through in 2011, and that wiped out many of the, or a lot of the SAV beds in the Hudson River, and some of these beds still have not recovered yet. So then, given all of those threats, what does it take to be resilient? In the background, you can actually see a Vallisneria americana bed with actually that white stuff there at the kind of at the top in the middle. That's actually, those are male flowers at the top of the, just floating at the top of the water surface. It's pretty neat. So there are 4 different options for populations. They can either acclimate, which means that they're somewhat tolerant of different conditions. So we can think of phenotypic plasticity maybe. They can also adapt, and that is genetically based, and that is, you need sexual reproduction for that, for this adaptation to occur, because you you have individuals recombining genetically. They can also relocate, and that can happen through natural dispersal with local restoration or assisted migration. And what's the difference here? Local restoration is where you take plants from a nearby bed, and then you can plant them with or without propagation in the greenhouse first into an area. Assisted migration actually comes from further away if you just don't have that local source. And then of course the fourth one is, well, that SAV bed might just have to die. Those individuals will die, and you're just not going to have an SAV bed there anymore. So today what I want to talk about is 3 projects, and then also tell you a little bit about what I do in my greenhouse in terms of propagation and my visions for getting genotypes back out into the wild, rewilding them. So the first thing is I like to just understand what we have out there. What is the diversity out there? When I came to the Appalachian Lab in 2000, I knew that I wanted to work with submerged aquatic plants because the Chesapeake Bay was right there in my backyard. But I also knew that there was a lot of restoration going on. But it wasn't clear to me whether anybody knew what the diversity was out there in terms of genetic diversity within Vallisneria, because Vallisneria was used quite a bit for restoration purposes. And so given that Vallisneria can reproduce asexually so well, it was very conceivable that the entire bay, the Chesapeake Bay, was just one huge super clone. And so if that's the case, it doesn't matter where plants come from and where they're put in, because it's basically one individual anyway. Another scenario is that they're just hugely diverse and that there's genetic structure. And if that's the case, then translocating from one area to another can have some major implications, because the plants might actually be maladapted. So I wanted to understand diversity. And then I also was interested in the impact of severe storms, especially Hurricane Lee. And then we just actually published a paper on that in 2023. And then I also want to touch on restoring the Croton River. So as I said, I started out in the Chesapeake Bay. And so we sampled, by that I mean, this is my long-term collaborator, Dr. Miley Neal at the University of Maryland College Park. She's actually the molecular ecologist on that. And this is a lot of her data. So I'm also representing her lab and all the diligent efforts that she's done. She's really amazing. So we sampled throughout the Chesapeake Bay. That's what all these dots are. And then what Miley has done is she's used a program called Structure to figure out which samples are more closely aligned with each other. And what she's actually found is that the bay, first of all, the Vallisneria americana in the bay is actually highly diverse. There's some beds that is just one clone, and other beds, when we sampled 30 individuals, every single individual was a different genotype. So it's not a super clone out there. It's highly diverse, so that was really cool to see. What was also really interesting is that the genetics in the Chesapeake Bay are quite structured. So the northern bay plants are genetically different from the central bay plants, which are actually exposed to higher salinities. And then in the Potomac, so those are the green and yellow dots, they are different from the Central Bay and the Northern Bay area. So there's non-tidal, the non-tidal Potomac is a bit different than the tidal Potomac, but there's also a lot of gene flow coming, especially from the top to the bottom. But anyway, there's basically 4 different types of Vallisneria, if you will. And so if you were to take samples or plants from the tidal Potomac and move them into, let's say, the Central Bay, that might not be the best idea. It would be prudent to actually try, and if you wanted to restore an area in the Central Bay, is to try and find plants from the Central Bay area. So this was really illuminating to really get a feel for, you know, what is the where should we source our plants from? We can do the same thing for the Hudson River. So this is the Hudson River here, and what we have on the left panel is just showing you the samples. The pink samples is what we sampled actually before Hurricane Lee, right before, and then we went actually back after the hurricane to actually in 2016, so a couple of years after the hurricane, to sample again. And we decided to sample more broadly, but also within the area that we sampled prior to the storms. And then on the right panel there, or basically in the middle of your screen, you actually see our diversity results. So the bigger bubbles means that the submerged aquatic plant bed, the Vallisneria bed, had higher diversity. So in general, there was actually higher diversity lower in the Hudson River than higher up, but it was actually, it was pretty variable. And then on the right-hand side of your screen, you actually see that structure diagram that you saw before for the Chesapeake Bay, just tipped on its head so that it aligns with the samples of the Hudson River. And so what you can see here is that the red samples, they correspond to the more saline habitats, and they are actually quite different from the samples further north, where you see quite a bit of intermixing. But what's really stark here is that the higher salinity areas do show differences in terms of their genetic composition. You can also do other analyses like barrier types of analyses, and that actually shows that there's more barriers down in the more saline habitats. So now I showed you some results from the Chesapeake Bay as well as from the Hudson River. We also, this is a PhD student, she also went up to the Kennebec River. So this is what you're seeing here. Left-hand panel is the Potomac River, middle is Hudson River, and the right panel is the Kennebec River. We had samples, actually 5 different beds per river system. That's what she did. This is Brittany Marsden West. And then she calculated the mean genotypic diversity across the different river systems. And what I just want to point out is that the Hudson River is actually lower in genotypic diversity than the other 2 rivers. And you can look at other genetic markers as well, and you see that actually the Hudson River is lower in diversity. So that was really interesting, and we wanted to ask why. Why is that occurring? I could go on actually for quite a while about this. We did publish, as I said, we did publish this work, just this latest work on the Hudson River in 2023. But what I just wanted to summarize is that we found relatively low genotypic and allelic diversity. The interesting thing is that it was already low in 2011 and didn't actually get lower after the storms. That was one of our hypotheses, that if you have a severe storm, like a severe disturbance, that would decrease diversity. But that was actually not the case on the entire, when you look at the river in its entirety, the diversity actually did not change, even though some beds were completely lost. So of course you have high diversity loss in some areas, but across the entire river, actually, we did not see that loss. And so there's two things that you can get from that. One of them is, oh, great, diversity was not lost. That's good news, right? But the other flip side of the coin is that we already had low genotypic and allelic diversity from the very beginning. So in a sense, it's sort of like this system gets disturbed enough times that diversity was already low before the storms. It's sort of like the ghost of other disturbances past. And so it's already pretty degraded. And so the question is, does diversity have to be augmented actually? We sampled a nursery and we only found one genotype in that nursery. This nursery is in the Hudson River, and people use this nursery for getting Vallisneria americana plants and getting them out in the field. But the thing is that, I guess I didn't mention that, it's a dioecious species. So there are male plants and female plants. This particular genotype in the nursery is a female plant and was very good at asexual reproduction, actually flowered really well too. But it's only one genotype, and without a male, it will never be able to reproduce. So that was an eye-opener. We found across the entire Hudson River that vegetative reproduction was actually quite common, and that we actually found not so much fruit production. And then that actually then makes sense that we didn't see so much diversity in the Hudson River compared to, let's say, the Chesapeake Bay, where we actually see a lot of fruit production there. So if you don't have as much fruit production, maybe it's because it's a cooler climate, then you have more asexual reproduction, which ends up forming bigger clones, and then therefore you have you sample less diversity that way. Good news is that there's no significant inbreeding, and we found that gene flow is indicated, and we see that in these high levels of differentiation among sites that yielded these 3 genetic populations. And we found, and I didn't show any of the results here, we detected 4 zones of vegetative dispersal. So we did see that we were able to find some genotypes in multiple different locations, which indicated vegetative dispersal. The role of adaptation versus dispersal limitation still remains to be determined. So we're still looking into the processes that are going on in the Hudson River. But a really interesting system, a lot to think about here. So another project I really quickly want to touch on is the Croton River. So first of all, I talked about Vallisneria across different rivers, and then focused in more on one river system, the Hudson River. And now I'm focusing on even closer on the Croton River. So hydrilla was discovered in the Croton River in 2013. So that's, that was an eye-opener. And it was really great actually for the Hudson River management community to see it, discover it relatively early, and try and make an impact there, try and manage it so that it doesn't go out of control, which is what it is actually in the Chesapeake Bay area. So a first year management plan was implemented in 2017. Why 4 years? It's because hydrilla needed to be monitored, and then a management plan needed to be designed, I guess. So it took a few years for it to be implemented. And then fluoridone was injected into the river at 2 to 5 parts per billion, which is a very low concentration. So the question now is, are non-target species impacted? We know that the Croton River has other submerged aquatic plants, and including Vallisneria americana. So what we did was we collected Vallisneria from 10 locations along the river and from 20 patches, basically 2 patchers per location. Then we genotyped 96 samples, and we propagated 168 of these plants. And then what we did was, because when we propagate, we propagate different genotypes and keep them separate, now we end up with replicates of the same genotype, and I'll talk about that in a little bit more. But what we could then do is do an experiment that subjected different genotypes to different herbicide concentrations. And so that's what you see here on the left panel. On the x-axis has herbicide concentration from 0, so that's our control, to 6 parts per billion. So that is actually higher than what the target concentration was in the field. And then on the y-axis, we have total chlorophyll. And what we see here is that even at starting at 4 parts per billion, but especially 5 and 6, we see a big decrease in total chlorophyll A concentrations in the leaf tissue. Okay, so there is a decline even at such low herbicide concentration. And so then the question is, what's the implication for survival? And that's what you see here on the right-hand side, where we have, we've tracked the alive plants that were able actually to survive the entire experiment, which was 9 weeks, and then dead. And so the way to interpret that is that, so going from top to bottom, you have the different herbicide concentration, including the control. And so if you have, it's a correlation plot. So if you have really big bubbles, that means that that it's a pretty strong correlation. And if it's blue, it's a positive correlation. And so what you can look at is, I don't know whether you see my pointer, but those large blue dots under the second column in concentrations 4, 5, and 6 are basically saying that in those concentrations, you actually see a whole lot more mortality in those units. And so essentially, the way to look at that is that, so you have chlorophyll A concentrations are lowered in leaf tissues starting in 4 parts per billion, and then especially in 6 parts per billion. And because of that lowering of chlorophyll A, you end up with less photosynthesis, of course, and then therefore higher mortality. So I thought that was really interesting. I looked at 9 different genotypes, and there was no difference in any of the genotypes. And maybe that's not so surprising. Why would there be any adaptation? Of course, there wouldn't be any adaptation to herbicides, but I was thinking maybe some of the genotypes might be a little bit more tolerant than others, but that is not the case. All genotypes were about equally tolerant to the herbicides. So then there are a bunch of different alternative approaches for active restoration. So I I should say, I should go back to this. So the Croton River, we looked at genetic diversity in the Croton River, and then we wanted to go back after herbicide went into the river, and then collect more samples, but we couldn't get any samples of Vallisneria because the plant population seemed to have crashed. And so we can't actually look at before and after. And so the question is, was it the herbicide? Was it something in the environment that affected the Vallisneria? We can't actually conclude anything from this. But unfortunately, we don't have a before and after genetic diversity. We would really love to have that. But given that Vallisneria isn't doing so well in the Croton River at this point, the question is, should we, given that we have propagated Croton genotypes, we have them in the greenhouse, we can actually bring them back into the Croton River. The same thing goes with the Hudson River in general. We've propagated from across the entire Hudson River, and so we could actually bring some of that propagated material back in. So I think it's really important then to think about alternative approaches for active restoration. So what you have there at the top is is this line where you can think of, is your goal just to represent genetic diversity as it is locally? So is there genetic diversity already in place locally? So that's on the left-hand side. On the right-hand side, we have the diversity is really degraded. Maybe the beds aren't even there yet. So we have to augment genetic diversity. So you have that spectrum of different goals. And so then if you just want to represent the genetic diversity that's there already, that's been measured already, you would want to select the stock. So here we're in the pink. We want to select the stock from local sites. And then we can move, as we move over to the right, towards needing to augment diversity, we can think about moving from within local areas. And if that stock is not available, then moving within regions. And then all the way to the right, we have managed relocations across regions, right? But what we want to do to be conservative is stay further to the left as much as possible. And if we don't have the stock, then we start thinking about moving to the right. We can think about perceived benefits. So if we select from local sites, we reduce the risk of genetic dilution and maladaptation. However, sometimes we need to have genetic rescue, so that's when we move within regions. And then if we really do have to move from outside the region, then we would want to match the stock to being adapted to those same conditions. There's of course risks, which are kind of the same. So that's in the red, that's very similar to the yellow. And then on the bottom in the blue, it gives you an idea of when we use what approach. So we would select from local sites when there is high diversity, low inbreeding, and also high differentiation, and also local adaptation if we know that. We move within regions if we have low diversity and high inbreeding. And then we would do managed relocations if persistence is just simply not possible because of climate change or the dispersal ability that precludes recolonization. So I wanted to just show you, this is actually a diagram that we came up with, that that restoration is not one size fits all, that we really need to understand the diversity of the system and what our goals are. And then that will determine how we select the stock. And that gets me to my Vallisneria americana repository. So we've sampled throughout the Chesapeake Bay as well as the Hudson River, collected hundreds of genotypes. We actually know the identity of the genotypes through microsatellite genetic analyses. And so what we then do, what I do is so what we do is we sample plants in the field, a whole plant, and then Miley only gets a little bit of the leaf tissue, and I get the rest of the plant, which I then plant into these little 5-quart containers at first, and then they then will asexually reproduce, right? So that after a few years, not even a few years, this can be just in 1 year, that whole bucket is filled with that same genotype, because that's just, you know, one clone. And so that's what you see here. You actually see what I'm in the back up here. You might see the label if you see my pointer, I don't know. SP stands for Stockport. So that's a Hudson Bay site. And then if you look really closely, you see that these are crotons right here. So we have croton plants and Stockport plants right in front of us. So what you can then do with this repository is you can do experiments where we can combine different genotypes into populations, or we can compare different genotypes and look at their traits. And they're all, it's amazing how different they are. They have different pigments. Some have wide leaves, some have narrow leaves. Some grow vigorously clonally, and others are very good at flowering. And so it's really amazing. They actually have different personalities to some extent. And so what you then can do is run experiments on them. And so this is actually what you see here. So all of these different labels here are different genotypes. These are actually from the Chesapeake Bay, but the Hudson River would have the same thing. And so you have on these different axes, you have some genotypes that have short leaves versus long leaves. You have some that have high tuber production. Some have very low tuber production. They are the real frustrating ones, because you just can never get enough material to actually do some high replication experiment. Then we have plants that, these actually go hand in hand. Low tuber production and few ramets go hand in hand. So you have few ramets versus many ramets, and so on. So you can, also the flowerers, but that actually goes deep into the graph here. So they are actually quite different, which was surprising to me that I could actually pick that up with some replication. And then I can actually combine different genotypes into populations, as I said. So on the x-axis, we have populations that only have 2 genotypes, 4 genotypes, and 8 genotypes. And then I looked at a bunch of different things, but one thing that really jumped out is the flowering part. And that is that when you have more genotypes in a system, you actually have more flowering events. So there's more chance of sexual reproduction and recombination, and therefore also the chance of adaptation to potentially different environmental conditions. So I thought that was really interesting. So in these experimental units, when I started them, the same number of individuals were added. The only difference was that for some of the individuals were the same in genotypic diversity of 2 versus genotypic diversity of 8. Each individual was actually different. So let me tell you a little bit about greenhouse propagation. This is actually a high school student, Sophia, who's helped me. What we like to do once, so I have this greenhouse repository, which tends to reside in the gray buckets, where each genotype has its own bucket. And that is so that we know we've identified this genotype and we don't want there to be any mixing between genotypes. But once it comes down to propagation, we want to, for the genotypes to have more room to grow. And that's when it's actually kind of nice to have them in these deeper water baths. And what's also nice is that you can now put a bunch of buckets into a big water bath. And some of those buckets will be female and some male, so that when they flower, they can recombine in the water baths. So these are my plant centurions. You've already seen them. So what we can do is if I know I want to propagate a genotype, I can just take the plants out in early summer, like in June. And so that's what we have here on the left-hand side. And then I replant them so that they have more room to grow, and I will replant them in different containers. Sometimes actually they will have turions attached to them. That's also kind of nice. That gives them more energy to grow. And sometimes I just go directly from turions. This is my setup. I've already showed you, we have the water baths here. I can fit 3 of these green buckets in, or actually 5 of the gray buckets. If you turn around in the greenhouse, you end up with this view. Okay, so we have a couple different types of containers here, depending on my goal, whether it's a repository, whether it's an experiment, or whether I'm trying to propagate. When we plant, what we do is, so I'm separating clones essentially. So I have a clone, I have a bucket filled with plants, and I take all the plants out and then I separate them and put them into several different containers. So that's what Sophia is doing right now in this picture. It's really fun for a high school student to do this. So that's the asexual reproduction part, but there's also the flowering part. Alex has already talked about that. So on the left-hand side, we have a female flower that is just emerging. In about 1 to 3 days, it will be completely open and on the water surface. So when you see these little nubbins, that's not the scientific term, but we have one right here and one actually emerging down here. We can be alerted to, okay, there's going to be a flower happening here soon. And then you have about 3 days for that flower to be pollinated before it just isn't viable anymore. And so here we actually see some male flowers on the surface, as we've already discussed. So then what do you do? Well, what you can actually do, this is really fun, you can pollinate. So if you have a bucket, a water bath, this one actually has 2 females and 1 male. There's actually, this is for Stockport. These are all Stockport genotypes. We have more females than males. So this bucket here, top left, is a female genotype, and you can actually see you see a bunch of different flowers. And then you see white string on some of them, and that means that they've been pollinated. You see a bunch of this sort of dandruffy looking material. That is the male flower, so the male also flowered. And so then it's pretty nice because you basically have this water bath, this soup and the male flowers will just find the female flowers. Usually I dip the female flowers into the male flower just to make sure. And then when I know that it's been pollinated, I actually tie a string around it. So what you can see here is a bunch of different pollination events that happened. In fact, actually on August 16, there were 5 female flowers. That's the genotype 863. The 863 is very prolific, a very prolific flower. And it was pollinated by the second genotype, which is 865, which is also very prolific. Then there are the other female flower, or other female genotype at 732. Not quite as prolific, but worked out too. And so here you can see a little bit more close up what's happening. So much fun. So sometimes a female will flower, but the male isn't flowering. I'm not seeing any male flowers on the surface. And so then hopefully a male flower from a different bucket will have done its thing. And so then I just take some of that flower, put it in a plastic cup, and then bring it over to the female flower. That's the beautiful thing about male flowers is that they just get released to the water surface. They're not actually attached to anything, whereas the female flowers, they stay put. And then we end up with seed pods that are probably about 2 inches, maybe even sometimes 3 inches long. And within each of these seed pods, you can have 100 to 200 seeds in them. So this is actually something that we've done just recently is we've harvested these seed pods. The nice thing about doing this in the greenhouse is that you don't have any waterfowl out there that will get to your seed pods before you do. And so for just one of the water baths, one of those black tubs, this is how many seed pods we were able to harvest. Overall, we harvested, I think, around 90 seed pods. This is just for one summer. So if you say times an average of 150 seeds per seed pod, you know, that's quite a few seeds here that we can put out into the wild, especially into Stockport, since these are coming from Stockport. And then we can also harvest the turions. So right around now, my technician, Jemima, she's an undergraduate at Frostburg State University. She harvests the tubs. And so what she does is we have a sieve here where she just takes a handful of the soil and she puts that in the sieve and then uses some water to just wash everything through. And it's super easy. You end up with all of these turions emerging. And then she just picks them out with her tweezers. I think we harvested, our record number of turions that we harvested was around 120 for a little one of those green buckets. Let me go, can I go back here? So one of these green buckets sometimes can yield 120 turions. The average is around 65 turions. That's a lot across all of these different buckets, and they're a nice size too. And then for every one of these buckets, all of these turions are the same genotype. We know that because we didn't really mix genotypes. And then what we can do in terms of restoration, I'm developing this methodology right now, is we can put these turions or plants, actually turions are better though, it'll be easier, into burlap bags. And then as the plants germinate, they emerge out of these burlap bags. And so on the left-hand side, that's actually cheesecloth, not burlap, but same sort of idea. They emerge out of the holes And then over several weeks, it doesn't take long. This is about 5 weeks after planting. You have a whole population of a whole clone, essentially. This is one genotype. So we planted 5, in this case plants, but they were small plants, into one burlap bag. And this is how much clonal growth happens out of these burlap bags. It's pretty remarkable. You can actually see here all of these different ramets forming, coming out. So the idea is these are just really easy to take out into the field, and then you can stake them into the ground so that they can stay put, and then they can continue on their clonal journey. And then if you have a bunch of different genotypes, this one has a pink ribbon because it's female. We also have male genotypes. And so if we plant these burlap bags of different female and male genotypes, we then can jumpstart a population of Vallisneria americana. And that's where I am actually right now with this, and I'm happy to take any questions. Thank you.
Thanks, Katja. So I'm checking out our chat box and I'm not seeing questions there. And I'm also looking in our question box. Let's see. If folks do have questions, whoops, we have please feel free to put them in the chat or the question box. Any questions for Alex as well? Okay, let's see if I can open this. Not a question, but wanted to say thank you for sharing all of this information about the exciting work happening, Brittany Rogers. And I wanted to ask a question if that's okay. So Katja, would it be possible if we were to collect samples of Vallisneria in the Croton River this coming season? Would that be possible then to do a pre and post treatment comparison?
Absolutely.
Okay.
Yeah, we love that. If you can find Vallisneria in the Croton River, we would love it. If we can come out, that would be great too, because that way we can make sure that the Vallisneria is collected in a way that would allow us to compare and us feeling confident about the comparison.
Okay, let's talk more offline.
Yep.
Then I have another question for Alex.
Yes.
As you are potentially heading to the Midwest again, are you planning to continue work on Vallisneria there?
That is a good question. I don't have any plans for employment at the moment.
Okay.
Still looking. We're about 6 months out, so I'm uncertain what I'm going to be doing. I'd love to continue working on aquatic plant research, though.
Okay. I'm keeping that, duly noting that information, Alex.
Yeah, I especially, like I highlighted that I would love to do a biogeographical tour of the US to capture different genotypes within the United States, just to be able to see if we can track down that that species boundary to see where, you know, we stop seeing Vallisneria neotropicalis and we start seeing, you know, the potential hybrids between neotropicalis and americana, and then where we start seeing americana popping up. So that'd be something I'd be really interested in doing.
Oh, we're totally interested in that too. I think you should definitely do that.
So were you,
that sounds great.
and my question is you had noted that you're using the microsatellites to track down south. Were you working with Dr. Sia Bongorum out of Florida?
That doesn't ring a bell, although we've
Okay.
been in contact with people Oh, yeah, yeah, we have. We did. We were connected with her.
Okay.
Yeah. I don't know where that's gone though.
It was an idea that was thrown out there that we wanted to try to collaborate on a project. But
Right.
since it wasn't Florida specific, it was tough to collaborate on a project that was outside of Florida.
Ah, okay.
Okay,
Yeah,
go ahead, sorry.
I was gonna say just that is such an interesting question where that species boundary is, whether hybridization is occurring. So I would love to think about that more. That'd be great. But yeah, it's hard. It's hard to get state funding or funding from a state because that crosses state boundaries.
Yeah.
So we have two more questions. We have Lindsay Yoder, who is our Region 9 New York State DEC AIS coordinator, said she is also interested in doing genotyping fellows for the Vallisneria in the Niagara River. So we can talk more about that too. Yay.
Yes, hi
And then
Lindsay. I need to get back to you.
And then we have a question from Mark Heilman from CPRO. He wanted to know if your data is available in a report that he can see.
It is.
Okay.
So I can send it to him, or I think Kathy, you have it too.
I'd have to dig and see.
But I'm happy to connect with you and share that report. And I'm still working on a paper.
Okay. Yeah, I think that's actually what Mark may be referring to. He had asked me about that, whether there's going to be a publication.
Yeah.
But he's busy typing, so let's give him a moment. He says thanks, paper or report will be good. Both are fine.
The reason why actually this paper is long in coming out is that it would be so much better if we had the before and after genetic diversity. And so crafting a story without that is different from if you actually had that information. I'd love to have it.
Okay. Okay, no pressure. We got it. All right, so I will touch base with you and our Region 3 coordinator, whom I think you may have interacted with, Ashley Morris. Maybe not. Okay. We can certainly talk more.
Maybe I have, yeah.
Okay. So anyone else have any questions? Please put them in the question box or the chat box. I'm not seeing any. Oh, we have another one. Yes, we should coordinate. Ashley's here. She said yes. Okay, good.
Hi, Ashley.
Okay. Well, seeing no more questions, I will say thank you both very much for very informative talks, and I'm really grateful that you're both doing this work. It's really, really helpful for us because we're heading into restoration work as well, and also very informative for the members of the society who are involved in restoration and aquatic invasive species control. So thank you again for taking the time to do this. I really appreciate it. And I wish everyone a wonderful rest of the day. Thank you very much. Take care. Bye.